today a little bit about Genomes to People, the program that I run in translational genomics and health outcomes. And I'm going to talk to you about the last mile problem, and I'm going to end up with what I'm most passionate about right now, which is understanding genomic penetrance, and hopefully give you some insight into some of the issues of the day. And perhaps you're wondering why we're not using genomics right now more in medicine and what we could do to do that. Academic life is supposed to be sometimes the honest broker in terms of people's enthusiasm, so it is important for me to make you aware of my disclosures. And I have to tell you that I'm here sort of sandwiched in between one big data talk yesterday and more big data talks this afternoon. I've got a little bit of big data envy here. It's not just that it's so cool what they're talking about in terms of scraping data and creating new inferences, creating new opportunities, but let's face it, they have the coolest slides ever. So I'm actually not going to talk to you about big data, but talk to you about what I think needs to balance big data in the realm of scientific inquiry. And that is a form of small data, where we estimate and empirically measure clinical utility. And that's because we've got to get this right when we're facing patients, as we do, one-to-one. And some people have called this the last mile problem, where you've got all this extraordinary technology and you've got the opportunity to utilize it, and somehow you've got to actually get it to the end user in a way that's appropriate. So let's start off talking about clinical utility. Clinical utility can be defined in lots of ways. I'm just curious about my information. That's one kind of personal utility. I might be planning a family. I might be taking certain types of drugs. I might be wanting to maximize my health, or I might be wanting to treat my cancer. All different types of clinical utility. The ones that are about actual specific medical benefits we think of as a narrow sense definition of clinical utility. And we can sort of think of broader senses that like concentric circles of an onion that are moving out from that. And the big data investigators among us, and there are lots of them today, are going to be very good at asking some types of questions, the learning health systems and all. But those types of inquiries are potentially vulnerable to certain types of bias in the way you ask those questions and the way you measure your results. On the other hand, there are slower, more conventional, more rigorous forms of inquiry, such as randomized clinical trials, that perhaps can give insights that are slightly more on target for making the pathway along that last mile. My program, which involves many, many scientists and many, many collaborations, is called Genomes to People, and we focus on medical, behavioral, and economic outcomes of genomic information. And this is complicated because genomicists and geneticists are probably the only people who are actually afraid of their own technology. And there is a strong tradition in medical genetics of being concerned about the downstream impact of genomic information. Much of this is well-founded. I don't mean to be cavalier about it, but it is interesting that the first impulse of so many people is to look at the negative side of genetic information. Now, we confronted this head-on starting in 2000 with the REVEAL study, where we looked to disclose information on people about their risk of Alzheimer's disease. How many of you know about your own ApoE risk, which gives you an estimate of your risk of developing Alzheimer's disease? Put your hand real high. How many of you would like to know about that? This is a very future-forward audience. Would you say the same if I had a phlebotomist at the door, I wonder? But nevertheless, it turns out a lot of people do. And we looked at this in a randomized clinical trial. We actually randomized people into learning about their ApoE, their genetic risk for probabilistic estimates of developing Alzheimer's disease, and not getting that information. And we demonstrated that, in fact, people were able to manage this information quite readily. So this was called the REVEAL study. It turns out that 24% of you probably have at least one copy of that E4 allele. And in our results, we demonstrated that people were no more likely to get distress, anxiety, depression. They were actually motivated to make changes in their lifestyle in terms of medications, exercise, diet. And in the first of its kind, they reported they were more likely, if they were E4 positive, to purchase long-term care insurance. This is called adverse selection, and let me tell you, it terrifies the insurance companies. A very interesting phenomenon going on with genomics and insurance. We were also the first to do a real in-depth look, prospectively, at the customers of direct-to-consumer genetic companies. Now, these are folks who are getting not just one genetic marker like ApoE, but getting dozens or even hundreds of genetic markers for risk of common complex diseases. And we looked at about 2,000 of these customers at various points in time and asked, why do they want the information? What did they do with it? Did they actually understand what they were getting? And what were the downstream psychological, behavioral, and economic outcomes? And I'll just tell you a few hints from this, which are that there are a lot of concerns, including the FDA, about people misunderstanding and misusing the information. Turns out that at least in the data we gathered, they understood the information really well. And when you worry about them actually changing their prescription medications without the permission of their doctor, almost none of them reported that they did that. And when you worry about them doing the jelly donut effect, you know the jelly donut effect, you tell me that I've got low genetic risk for diabetes, and I tell you, pass me the jelly donut? A kind of false reassurance that people might accrue by assuming that their genetic risk is really more deterministic than it is. Well, it turns out that people don't ascribe to that. At least the customers at this early point in time all had a kind of halo effect, in fact, where they were more likely to report that they were improving their diet, they were improving their exercise, even though it wasn't always linked to the kind of risks they were getting. But now we're in the era of genomic sequencing, whole genome and whole exome sequencing. And the question that I'm obsessed with right now is, how will we use exome and genome sequencing in the everyday practice of medicine? Now with this, of course, we're sort of moving from the common, complex, modest effect size space that we were just talking about with ApoE and direct-to-consumer genetic testing into the Mendelian space, the more deterministic, rarer Mendelian diseases, the dominant and recessive conditions that we have always had in traditional genetics. And as we start using genomic sequencing in medicine, the first problem we had to confront was the problem, or you might think of it the opportunity, of incidental findings. Because when you do a whole genome for whatever reason, let's say for your heart, you're likely to find something else. That's, for example, you have a cancer predisposition, and there was all sorts of machinations about, do people want to know? Is it appropriate to disclose? Does it mean the same thing if you find it in an incidental way as if you find it when you're looking for it? And so we have written a summary article about the entire process of sequencing for diagnostic purposes, but we also have led an initiative to create a policy around incidental findings, because frankly, the field was in some chaos about this. And what we basically said was, as a starting point, when any laboratory does any kind of sequence, you should be reporting back on 56 genes. Now, with 20,000 genes and about 5,000 disease-associated genes, I'm imagining this audience probably thinks that's pretty paltry. But believe me, getting those 56 genes together and getting some kind of consensus on them was the hardest thing that I and my colleagues on this working group have ever done. The 56 genes represent 24 conditions, which are mostly inherited cancer disorders and inherited cardiac disorders. And they're present, pathogenic variants or real mutations in these genes are present in about 1% of people. So about 6 people in this audience, if you're all here, are actually walking around with a rare Mendelian mutation in a cancer or cardiac disease predisposition gene that you don't know about. That's really interesting. But would it make a difference? And that's a really hard question to answer. In fact, the whole thing is conflated with what's the right analogy here. So for example, if this is California, we all ride our bikes. If you fall off your bike, you go to the emergency room, you get a chest x-ray to check and see if you've got a broken rib, and the radiologist spots a shadow on your lung. The radiologist doesn't sit down with you and say, hey, you know, we could find something, would you want to know? Let's do some counseling about it. No, they report the finding back to your clinician. They have a fiduciary duty to do that. It goes in your medical record, and your clinician will contextualize that by saying, oh, are you a smoker? Where have you traveled? And so forth. But now we're thinking about genomics, and genomics can cast a much broader net. And people are quite concerned, appropriately, that this harkens back to when we would throw people into whole body scanners, and we'd find all sorts of things, and there'd be a lot of incidental and false positive findings that could take money and even harm in working them up. So we've got the MedSeek project funded, thanks to your tax dollars, and we are actually asking what would happen if you did a randomized trial of using whole genome sequence in the everyday practice of medicine. We've taken two groups, a group of patients that actually have a disease that has a genetic component, cardiomyopathy, and more controversially, a group of ostensibly healthy individuals, and we're randomizing both those groups into standard of care and whole genome sequencing. And then, with incredible granularity, we're following what happens in those interactions, what happens in the medical record, and what happens to those patients when they get this information. And let me give you a few early insights into the data we're collecting. First of all, this is harder to sell than you might imagine. Turns out that people are really concerned about genetic discrimination. Even though GINA protects you from some forms, it doesn't protect you from all forms. And worry about genetic discrimination, insurance discrimination is the #1 reason why people got into the consent process and then declined to continue with the study. So we've got a real issue in this country with insurance discrimination that we still need to solve. #2, it is not easy to characterize these variants. We all have 4 million variants. We all have hundreds of variants in our disease-associated genes. We can automate part of this process, but part of it is manually curated even now. And we're working on ways to share these data so that this process gets more efficient, but right now it's pretty hairy. And the other innovation we did, though it's not too flashy, is that we created a one-page summary of your whole genome sequence for a primary care doctor. Now, many people thought this could not actually be done, but we're showing that our primary care doctors are using this one-page summary to do sensible contextualization of genomic information. And it shows you your deterministic dominant conditions, your recessive carrier traits, your pharmacogenomic variants, and even a section we've innovated on in terms of determining your blood group through your whole genome sequence. Now, here's what's startling about this. We're finding that over 90% of people have a recessive carrier trait, sometimes several. Okay, actually you knew that. That's not too startling. But here's what's really startling. About 20 people out of 100, when we go through this very elaborate variant classification process, have a pathogenic or likely pathogenic variant for a dominant genetic condition. And folks, they don't have that disease. What's going on here? Turns like 20% of you are walking around with a real mutation, and you probably don't have the disease. Or if you do, you don't have it yet. And which of those is it? Because that's the $24 million question. We can't be assuming that everyone who has a mutation has the disease. That's 100% penetrance. We know that's not true for these genetic variants. How are we going to figure out what the penetrance really is? Well, it's really important because when you give this information to doctors, they do what doctors do, right? They order stuff. And in fact, our primary care doctors are ordering very sensible things when they get this information on those one-page summaries. And that's going to drive up the cost of healthcare. There's no question about it. In fact, what we really have to ask is, are we making some sort of difference in early disease prevention and detection versus a cascade of harmful and deleterious and distracting effects? What's the cost of that, and how can we calculate some sort of cost effectiveness so we can get at the value of genomic sequencing and how we implement that value in the practice of medicine? That's harder than it looks, in part because the clinical utility may not be apparent for decades. Well, how are we starting to do this? We are looking at this in the MedSeek project in this fine-grained way and comparing the two randomized arms in terms of the time spent with the doctor. Clearly, you spend more time if you've got to talk about a sequence. And in the economic costs associated with referrals and lab tests. Clearly, it's higher. But how can we get at the medical benefit when we can't practically follow these people for another 2 or 3 decades? How do we measure penetrance? Well, I'm going to show you some very, very early data that I'm really excited about. And this is the famous Framingham heart study. In the Framingham heart study, we've been looking at a publicly available database of nearly 500 individuals who are exome sequenced and who've been followed for an average of 20 years. So they've had time to manifest the manifestations of their mutations. And we only found 5 people, I told you it was about 1%, we only found 5 people who were carrying one of these mutations. And it turns out if you follow them for 20 years, 4 out of 5 of those people did develop some form of clinical feature that could be linked to that mutation. And if you model that in the rest of the people who don't have those mutations, it's highly significant. Small numbers, but highly significant. And we're repeating this right now in other larger scale venues. So preliminary data, very small sample size, it's very disruptive to the notion of genomic penetrance because we're talking about aggregate penetrance. We're not talking about the penetrance of a single gene or a single variant. And if you believe that screening could be beneficial for adults, and there's so many childhood onset pediatric diseases, right? Then you're probably thinking it would be even better for babies. And indeed, we just got funding from NIH to initiate the BabySeq project, where we're doing the same sort of thing in newborn babies. And let me tell you, this is a challenge from the IRB to the parents to the doctors on the ward. This is a difficult study to run. But perhaps if I come here again, I'll tell you some of our early insights from that. I will say that people are frightened of genomic information. And it's up to us to demonstrate to them that it can be provided with fairness, without hype, with an awareness of the harms and the benefits, and with real evidence behind all that. Last slide, I just want to say that in the spirit of kind of collaborative work, we've started a consortium we call the PeopleSeq Consortium of all those healthy people out there who are getting sequenced. How many people in this room have personally been sequenced? Wow, quite a few. And I'm sure many of you are looking to get sequenced. Well, there are a number of centers that are doing this either clinically or as part of research studies. We're actually getting us all to work together in a very democratic consortium to share the kind of outcomes that are taking place if you get sequenced. And I'd like to invite any of you who are running sequencing for ostensibly healthy people or who are getting sequenced yourself to contact us and be part of the PeopleSeq Consortium research. I want to finish there, but thank you very much. I want to mention as I finish that we are excited to collaborate with you. We'd love people who are interested in higher tech ways of moving this forward and collecting better empirical evidence. And we're also recruiting for some high-level leadership positions in genetic counselors, project managers, and research assistants in our most high-profile projects, including the BabySeq project. So if you know people that would be interested in that, please send them our way. I'd love to hear from you, and I'd love to talk to you more about these and your issues throughout the conference. Thank you very much.
Robert, thank you. Thank you. And let me ask you one question here. This is terrific. And so many of us have had our genomes done, and yet our doctors aren't playing in this game. What do we do in the meantime?
Well, it's absolutely true that regular doctors are unfamiliar and uncomfortable with this information. So I think there are more and more medical geneticists who are starting to be comfortable with this. There's more and more genetic counselors who are starting to be comfortable with this. And you can seek out one of the medical centers where they are returning this information and work with them to get your results back.
See you on Biotech Nation. Thank you, Robert. Thank you, Robert.